Medicina, en nombre del personal académico, administrativo, manual y cuerpo directivo de nuestra dependencia, agradecemos su participación en cada uno de nuestros eventos. Aprovecho esta oportunidad para pedir nuevamente a todos los asistentes que mantengan apagados sus micrófonos y sus cámaras a fin de poder asegurar la calidad de la transmisión. De igual forma, en sus pantallas podrán encontrar el chat, el cual estará activado durante toda la sesión, en el cual podrán escribir las preguntas y comentarios que quisieran hacerle al doctor Reza Nassiri, las cuales serán respondidas al final de la presentación. El día de hoy contaremos con la presencia de un invitado de honor, quien en el mes de octubre pasado estuvo en este mismo espacio compartiendo conocimientos, experiencias y opiniones sobre los retos y perspectivas del COVID-19. El día de hoy, para el webinar del día, el doctor Pablo González nos estará apoyando con la participación en actividades de interpretación para la sección de preguntas y respuestas que haremos al final. Pero antes de iniciar, quisiera ceder la palabra a nuestro director, el doctor Carlos Castro Sansores, quien nos dará unas breves palabras de bienvenida. Adelante, doctor Castro.
Well, thank you. Buenos días a todos. Morning, doctor Nassiri. Gracias a todos por su interés y su participación en nuestro webinar, que en esta ocasión se engalana con la presencia del doctor Reza Nassiri y tan importante tema que hoy está en boca de todos en nuestro medio en relación a las vacunas para el COVID. Le agradecemos al doctor Nassiri, como siempre, su atención, su disponibilidad y su apoyo para hacer llegar la ciencia y el conocimiento al mayor número de personas y profesionales posibles. Muchas gracias a todos por su presencia. Muchas gracias, doctor Nassiri. Y bueno, lo dejamos entonces con el micrófono. Gracias.
Thank you.
Muchas gracias, doctor Castro, por sus palabras. La comunidad universitaria agradece los esfuerzos por promover espacios de actualización de todos los profesionales de la salud, particularmente en los tiempos actuales, en los que los profesionales requieren de los conocimientos más recientes para afrontar los problemas de salud de nuestro país. Y es por ello que el día de hoy nos honra poder contar con la presencia del doctor Reza Nassiri, quien actualmente es profesor de farmacología clínica, profesor de medicina familiar y comunitaria, profesor de salud global, enfermedades infecciosas y medicina tropical en la Facultad de Medicina Osteopática de la Universidad Estatal de Michigan. Sus intereses de investigación se centran en la farmacología clínica del VIH-Sida y de la tuberculosis, la prevención y control de enfermedades infecciosas, enfermedades tropicales desatendidas, salud comunitaria, salud global y determinantes socioéticos de la salud. El profesor Nassiri trabaja en temas de salud pública internacional y tiene experiencia en educación, investigación, políticas y gobernanza en salud global. Ha realizado contribuciones en varios campos de las ciencias médicas, incluida la investigación clínica y la educación para la salud. Sobre la base de su amplia experiencia y conocimientos en VIH, SIDA y tuberculosis, desarrolló programas de investigación clínica en Brasil, Sudáfrica, Haití, República Dominicana y México. Los focos centrales de tales programas son el determinante sociocultural y bioético de la prevención, el control y la intervención del VIH-Sida y la tuberculosis. Todo lo anterior es un breve resumen de la amplia experiencia y conocimientos con los que el doctor Reza Nassiri cuenta y que el día de hoy nos compartirá en el webinar Las plataformas de las vacunas COVID-19. Adelante, doctor.
Thank you. Thank you. Can I go back to slides? Can you show slides? Yes. Thank you. Well, thank you so much for your very kind introduction. Let me acknowledge UADY School of Medicine, the Dean, Dr. Castro, and also Dr. Garcia, Dr. Gonzalez, and the doctor who just introduced me. I'm sorry, I don't know his name, and also the whole IT team. It's a pleasure to be back, and this time talking about vaccine platforms. To begin with, Let me just go fundamentally over the virus structure. As you know, it's SARS-CoV-2 is an RNA virus. It's a single RNA genome, and it has 29,674 nucleotide base pair. It has 2 open reading frames, okay? And there are some envelope proteins, membrane proteins, but most importantly, this spike protein, which serves as a receptor for the virus and binds to human ACE2, that's the subject of vaccine development, S protein. As you can see, the protein itself, that's the three-dimensional structure, in the eyes of biochemists. I'm not a biochemist. I'm a French-trained hematologist and with clinical pharmacology training. So you can see that it's a tripeptide, okay? In other words, it has 3 protein units. In yellow color are the sugar. So it's interesting that this protein also has sugar moieties. In addition, there are 3 proteins, the blue color and the orange color, and also the green color. And this is very important, actually, in vaccine immunogenicity, which I'll be talking about a little bit later, but also in vaccine resistance, the mutations that occur very frequently. If I have time, I will address it. If not, then we can postpone it to other times so we could discuss the mutations as well. So right now, this S protein, okay, with its sugar moieties is the subject of vaccine platform. And keep in mind, these sugar moieties here not only provides stability to the S protein, but also they are very vital in viral replication. So vaccine research and development, it's a very daunting effort. There are a lot of things aside from science goes into it. This is the slide. I am author of this slide. I developed it myself. So in vaccine development, the most important item is ethical considerations, okay? Where the vaccine was tested, for example, on what population was tested, whether these vaccines tested, they had authorities or license to go to different population or not, okay? So in ethical considerations, you know, there is efficacy. I'll be talking about that and what are the potential adverse effects, all right? And then we have immunogenicity and reactogenicity. These 2 are also integral part of research and development, but keep in mind when it comes to immunogenicity and reactogenicity, you know, one must have manufacturing quality control. So manufacturing quality control is a non-science aspect of vaccine development. Oftentimes it is quite ignored. We always talk about the science, but we also need to know what are the technology they're using and what are the pitfalls and pros and cons of that particular technology in vaccine, you know, development and subsequently delivering. So whether it's the assessment of the efficacy or adverse effects or immunogenicity or reactogenicity, we are all concerned from here all the way here, risk versus benefit. Otherwise, you know, it won't serve the purpose. And the risk versus benefit concept is very important for FDA, US FDA, whether they are capable of, allowing, authorization or not. But when it comes to immunogenicity, there are specific tests. They must do it. One would be the antibody test. The other one is assessment of especially cytotoxic T cells. I'll be addressing, in the next slides. So there are 3 phases of vaccine development, but keep in mind there's also another phase, which is phase 4, is involved, and I'll explain phase 4. The phase 1 basically engages a very small group, and the intent is only safety. You want to assess safety. So maybe you can ask, why is it a small group? Well, because we don't know much about vaccine. We don't know much about the safety profile. So we deliberately choose a small group if in case there is something that is quite, acute and necessitates intervention, hospitalization. So we're able to handle this small group. Whereas if we give it phase 1 to large group, then if in case there are some disastrous adverse reactions, it will be very difficult, to control it. So phase 1 starts with a small group and strictly the safety, not the efficacy. Then we come to the phase 2, which has more number of participants, could be hundreds, it could go even, thousands. Phase 2 is extremely essential because not only it further assesses the safety, but also it is going to establish the efficacy and immunogenicity, okay? In addition, the timing and the amount of, the vaccine should be administered. Therefore, phase 2 is fundamental, but also extremely critical. Then we have phase 3, which is almost the end. There are more participants involved in phase 3, thousands, so many thousands, okay, or even hundreds of thousands. And basically, we're looking into 2 confounders in phase 3. One would be, to assess detail, adverse effects. The other one would be, the efficacy. But keep in mind, once vaccine is temporarily approved by FDA, it has to go to general population. It is only through that stage, we call it phase 4, and we make further assessment of general population, because phase 1, phase 2, phase 3 are not considered to be general population. They are selected population. Whereas phase 4 goes to anywhere, every ethnicity, gender, so many confounders place. So this is the real assessment. I was talking to Dr. Gonzalez about it, so Mexico needs to follow this assessment. And for Mexico, particularly for Yucatan, my advice is to use as many different as platform you can, because that could give you a broader, not only perspective in choosing the right vaccine, but then ultimately by choosing at least 2 or 3 different vaccines, at the end of this year, you can come up with scientific assessment and evaluation that which one of those platforms serves better your population. So this is the hardcore area. There are so many platforms and there are so many ways of calculating the efficacy, okay? And it could be a little bit tricky, depends on the biostatisticians in every pharmaceutical company. And when I go through different slides, I will alert you the flaws of some of those results obtained from vaccine research and development. But basically, we calculate the attack rate, okay? That is the most important denominator in determining the vaccine efficacy. And let us take a very simple example. So you give a vaccine, and in your vaccine arm, 5% they come up with COVID. So, but in your placebo arm, 40% they come up with COVID. So there's a factor of 8 involved here. So on a simple calculation, 5% divided by 40%, that will give you 12.5%. Okay, so if you simply subtract this 12.5% from 100%, that will give you an efficacy rate of, let us say, 87.5%. That is the simplest calculation, although, you know, it could go very complicated depending on pharmaceutical companies, how they can manipulate these data, and this is a large area of discussion which is beyond the capacity of my COVID presentation today. So the question is, what are we looking for as endpoint? In other words, if the vaccine is able to prevent disease, if the vaccine is able to prevent infection, if the vaccine is capable of preventing transmission, is the vaccine capable of preventing hospitalization? Is the vaccine capable of preventing ICU? Is the vaccine capable of preventing ventilation? Is the vaccine capable of finally preventing death? So you see, there are numerous denominators involved in vaccine efficacy. It's not as simple as we most often think about it. You know, it has to be based on certain immunological and scientific and clinical rationale. So if you ask me, where do we stand now? Well, right now, we just started vaccination. But we had, prior to vaccination, some clinical trials, at least Moderna and Pfizer, okay? So that was a few months ago. Plus now we are on the 3rd month of vaccination, okay? So right now we are at the disease stage. We know that vaccines so far, up to date, this date, can prevent disease. But honestly, on a scientific basis, we have no idea today about the exact number for infection, preventing infection, and exact number for preventing transmission. So some of you may know that last week there was a report, not a scientific paper report, from Israel, and they did a preliminary assessment of vaccine efficacy in their population. As you know, Israel is #1 in vaccination strategies, and they concluded Pfizer is capable of preventing the disease, okay, by 98%. Well, that's good, okay? Although in one of the reports they said infection, but it's pretty immature to say infection. It takes us probably another year to fully make assessment or fully come up with the number for infection and transmission. But as of today, we know, for example, mRNA vaccines, at least from our experience in the United States, or for experience of Israel, you know, they're good. They have a very optimal efficacy, okay? But also keep in mind, when these vaccines, they go out in population, it really depends on age, depends on sex, ethnicity, comorbid condition, especially immunosuppression. So far, we do not have any concrete or conclusive data how these vaccines behave in immunocompromised states. So now, traditionally, a vaccine development will take years, you know, at least 7 years. But because of pandemic nature of SARS-CoV-2, we circumvented some of those steps. However, I should add, there were two advantages for this pandemic. One is the advances in molecular science, and the second one is the precision technology. I'm sure you see through your news network, they show all these vaccine factories, and they're mass producing, they're fully automated, fully computerized. Well, guess what? 50 years ago, we don't have this kind of luxury. As a matter of fact, if COVID came to us, let's put it this way, if COVID came to us 50 years ago, we would have been, the whole earth would have been devastated, because 50 years ago, we did not have advances in molecular science, okay, mRNA, or even recombinant DNA technology, which we use it for other, especially in cancer vaccinology as well, is a result of recent years, especially result of past 20 years of advancement, not only in our technology, I call it precision technology, but also advancement in molecular science. So think of this. If this had hit us 50 years ago, okay, it would have been really devastating. And a lot of people would have been dead on the spot. So yes, we circumvented the traditional approach and timeline, but there were two advantages in favor of us. One would have been the molecular science, the other one is the precision technology. So on this slide, no matter how you put it together, okay, there are four general categories of vaccine platform. One would be the viral vector. The other one is mRNA technology, which is quite new. The third one is, the whole virus or killed virus. And this one is the newest version, protein subunit. They mass produce the spike protein, especially in insects. I'll come to one slide about it, So no matter whether it's traditional viral vector, or novel technologies like RNA or protein subunit, or killed virus or whole virus, Chinese are using this technology, by the way. The end point is production of antibodies. And we must also make assessment whether these antibodies produced, are they neutralizing antibodies or are they non-neutralizing antibodies? Okay, it's very important. We just keep saying, antibodies. Well, it's good to say antibodies, but more precisely, we need to find out these antibodies are produced. Are they functional? Are they adequate? Are they neutralizing antibodies? Or it could be antibody-dependent enhancement, which can even make things worse. We have a couple of, cases about antibody enhancement, phenomenon after a couple individuals, they received vaccines. So that is very important to make assessment that whether these antibodies are functional, or not. So the other concept that I would like to introduce to yourself, and is the concept of immunogenicity. Okay, immunogenicity is an integral component of vaccine development. In immunogenicity assessment, okay, not only we need to make sure the antibody blocks the entry of virus into cells, we call it receptor domain blocker, okay? But also we have to make sure antibody produced by vaccines go into after circulating viruses. In other words, if a virus is circulating in interstitial space or in the blood, are those antibodies produced as a result of vaccination capable of going after the free virus, but also most importantly, antibodies that act at the receptor site. So for each one of them, there are different assays. You can't just say, yeah, virus works here, but how about here? A good vaccine is the type of vaccine addresses both in a very high efficiency. So I want you to take a note of it. If one step is missing, the other mistake is not effective. So it has to be both targets that could be, assessed. So one of the very common assays, I would say, in the past 5-6 years is luciferase assay. So in luciferase assay, you can choose a pseudovirus. These are basically lentiviruses. You can easily manipulate them. See, adenovirus, lentiviruses are easily manipulatable. You can manipulate them. You can make it genetically different, and that will give you a very good outcome. So these are basically pseudoassay, that's why it's lentivirus, but they manipulate it in a way that this virus is expressing S protein, okay? In addition, the genetic material of the virus is quite inert, but they add luciferase enzyme. So in a normal situation, in a vitro situation, you have this virus has a S protein combined to the ACE receptor in the cell. So the virus starts replicating here. The more virus replicates, the more luciferase, therefore more lights is contained, all right, is generated. On the other hand, in a test, okay, this is, let's say this is placebo, this is the real arm, test arm, in the presence of antibodies, so these neutralizing antibodies will not allow the virus to go inside the cell, therefore you have no signal. So here you have a very high signal, and here you have no signal. So what you do in that case, you put, it's all automation. This is the luciferase, automated machine. So it gives you the amount of light generated, then it calculates for you how much antibody, prevented the entry of the cells, and then you have the control as well. So then you can make a scientific calculation of the efficacy of the virus. So that was through the antibody arm. But keep in mind, in the last presentation I had, I emphasized on the role of T cells, okay? Specifically, there are two subsets, the helper T cells and cytotoxic T cells. The assays for T cells are not easy. Those of you who work in the lab, the researchers, and they're immunologists and they do assays with T cells, it's a very tedious, procedure. It must have a very good accuracy, and especially the reagents, So there's a lot goes into that, which is, I'm limited on time. Otherwise, I would have been happy to elaborate on that. So the assessment of both helper T cells and cytotoxic T cells, very important. However, I personally believe that cytotoxic T cells is fundamental for us, whether we recover from the COVID disease or whether we have been vaccinated and we have immunity. Either way, through our understanding, the cytotoxic T cells, they are very pivotal role they play, in defending, the virus. So that's a different topic. We can address it, later. So what happens? On this slide, there's an approval, okay? And this was published in a prestigious journal of Nature Medicine. I like it. It's very scientifically oriented. I wrote to the authors here, and I had one fundamental issue with this figure. Can you figure out what is the fundamental issue with this figure? I'll give you a few seconds. Well, this is excellent figure, all right? But for approval, it is not only the efficacy, it is also the safety. Although they address the safety here, but to begin with, okay, for approval, it's efficacy plus safety. So I wrote to them and I asked them to reorganize this figure. Then it comes to manufacturing. So I addressed the quality a little bit at the beginning of this webinar. You know, quality is extremely important. Okay. Now, I do not want to politicize this webinar, but there are certain countries, okay, the questions, answers you can ask me, you know, they may have the best science, and they do. But when it comes to manufacturing quality, it's always questionable, okay? So it's not the science, but also manufacturing is very important, you know. Then it goes into global vaccination programs, okay? After that, you know, you need to make assessment. That is basically the phase 4. From here to here was phase 3 results, then, you know, enters phase 4. And here in phase 4, there's a lot of confounders, you know, it could be age, ethnicity, you know, and even lower socioeconomic class, so on and so forth, you know, and who gets the vaccine first. One of my concerns over Mexico is the indigenous people. You know, those of you who know me, I used to be in Merida a lot. I had a lot of clinical collaboration there. I used to bring students there. And I have a high respect for indigenous Yucatan people, Mayan people, or let's say Chiapas, you know. So my fundamental question is that what is the overall policy of your health ministry, federal, Distrito Federal? Are they going to give these population, indigenous population, a preference to vaccinate them as soon as possible or not? That's a question I have, and at the end of this webinar, we can address that issue. Okay, so then we have to the concept of reactogenicity. These are basically adverse reactions. It could be in the setting of acute, or it could be in the setting of systemic adverse reactions. So what it is, you first get the injection into deltoid muscle, okay? Now, here I would like to emphasize, you really need to have experienced individuals to inject, okay? I have seen people, they go above deltoid muscle or below deltoid muscle. That is the wrong approach. But also, these are the people develop most adverse reactions. It has to be right in the center of deltoid muscle. I've seen it through several, you know, videos published by news network, you know, and I keep saying, wait a minute, you know, who taught them vaccination? Who taught them vaccination? So it's not enough to train a person for a couple hours of injecting vaccine and send that person out for vaccination. That is actually unethical, okay? Another word, a person is injecting above or below, there is also ethical issues involved, in addition to competency issue. So make sure the person is injecting a vaccine in Merida or Yucatan, you know, they have adequate training. Okay, so by way of immunological reaction, you get, you know, side effect. It could be erythema. It could be, you know, different redness. Okay, excuse me. Then once, you know, the vaccine is systemic, then it can provoke certain production of cytokines, you know, and then once cytokines are produced, or chemokines, you know, you will see that C-reactive protein goes a little bit up, Okay, so if the C-reactive protein goes a little bit up, don't worry about it. It will eventually come down, you know. Then the individual, your patient develops fever. That happens usually during the second dose. Okay, first dose doesn't happen much. Okay, the second dose. So here on the second dose, when they develop these symptoms, my recommendation is if these reactions, reactogenic reactions, is within 24 hours post-vaccination, that's fine. But if the patient is experiencing symptoms related to vaccine for more than 2 days, then my recommendation is that that patient who received vaccine needs clinical evaluation and possibly hospitalization. Okay? So in that context, let's go to the next slide. I can elaborate a little bit more. So after receiving a vaccine, there's always potential that you develop inflammation. Okay? And this is through specific receptors called pattern recognition receptors. Okay? Pattern recognition receptors. And these pattern recognition receptors are associated with inflammation. So the patient may get an inflammatory response. So you may ask them, what's the difference between antigen, vaccine, and the active disease? Well, the active disease has live virus. So the live virus is continuously duplicating. Okay, continuously using these ACE receptors, okay, in the body and duplicating. So the more duplication means the more inflammatory reaction, okay, and the more inflammatory reaction can go even to extreme scenario, which could cause hyperinflammatory response. So that is the virus scenario, whereas the vaccine scenario, because it's only antigen, okay, it doesn't have to duplicate. So the extent of inflammation is limited. That's the key point. The extent of inflammation is limited, and even the duration of inflammation is limited, unless there are few cases in the United States, but that's a different mechanism, immunogenic mechanism, which if I have time during question and answer, you know, I'll be happy to address that issue. Eventually, there are mast cells, monocytes, macrophage, they're activated. They produce chemokines, cytokines. There is inflammatory reactions. It could be circulatory inflammatory reactions or local inflammatory reactions, but eventually, because the vaccine is not the live virus, okay, so it cannot continue replicating. It's just an antigen. Antigen is not capable of replication. Therefore, it will stop at some point. So then what are the critical factors? Okay, well, it could be the vaccine factor itself. One of the vaccine factor is polyethylene glycol. We call it PEG. Well, polyethylene glycol is immunogenic, okay, especially in some susceptible individuals, you know, can cause immunogenic reaction. We know from our literature 60 to 70% may develop antibodies, you know, to PEG, okay? The other aspect of vaccine factor is, as we have seen in just few individuals, not many, it can produce, and these are basically individuals, they have immunogenetic predisposition, okay, not in normal individuals. It can produce non-neutralizing antibodies, okay? At the beginning of my webinar presentation, I mentioned it, leading to ADE. So that could even worsen, you know, the situation. But then there are, you know, administration factor. Really, the person should be experienced. You know, when it says deltoid muscle, it should not be above deltoid muscle, the injection, or it should not be below. It should be right in the center of deltoid muscle, okay? And then there are other genetics, you know, the race and gender, ethnicity, especially body mass, you know, and the body mass index, you know, they all play into vaccine efficacy. So the question here is, what is the difference between efficacy and effectiveness? Well, efficacy is simply the measurement of how vaccine works during clinical trials, okay? Whereas effectiveness, okay, is the type of measurements goes into phase 4 evaluation. Remember that general population, okay? So we are now just the 2.5 months started to evaluate, you know, phase 4, although it's too early. I would say in the midsummer, we should start compiling our data, see how the vaccine performance was with respect to different population, ethnicity, socioeconomic studies. So many confounders goes into that, okay? So we know for sure that there is no vaccine, even Moderna and Pfizer, they don't have 100% efficacy. It's almost impossible, especially if it comes to immunosuppression cases, you know. But from our retrospective study, let's say measles, okay, we know efficacy measles varies a little bit between 95 and 98%, you know. However, you know, on average, measles is only effective in 96% of individuals. That means 4% may not benefit from measles vaccine, okay? Now, 4% may sound a small number, but when it comes to population, let us say 4% of 350 million population, United States, that's a lot of number, okay? So all I'm saying on this slide, you know, there are some small percentages may not benefit from vaccine at all. So what are the key points that I talked so far? Well, most vaccines so far we know they have not addressed the asymptomatic and pre-symptomatics, okay? The success of our program, which started about 2.5 months ago, really depends, you know, how efficient are the program, okay? It's not only vaccine itself. It is about how efficient and how prompt is your team that delivering the vaccine, and how efficient is your resources, especially storage resources, distribution, okay? And then vaccine effectiveness, we just started evaluating effectiveness. Keep in mind, this is not efficacy, this effectiveness. But if you put them all together, the vaccine development for COVID was really a remarkable science, to my opinion. Okay. Well, Dr. Gonzalez told me he received, I think, Pfizer. All right. Well, here's the Pfizer data. All right. So they established efficacy in vaccine arm versus placebo arm. This is the cumulative data. So you can see in placebo arm, these dots, dark dots, these are the patients develop severe infection. Look at here, these two in this vaccine arm, okay? Although I don't understand why they put one, you know, there should be few, but anyway, they demonstrated the efficacy very nicely. And this was a type of vaccine trial, okay, that had 43,548 participants, age 68, all the way for Pfizer was 71, okay? Now, the good thing that I personally appreciate about this kind of data, you can see the vaccine arm. I don't know how big you see it. You know, on my screen, this slide is very small. I'm actually having trouble seeing it. You know, it's not a full screen for me. But anyway, you can see that on vaccine arm, the number is almost equal to placebo arm. That is excellent. Okay, in other words, there is no dichotomy here. Confounders are eliminated. So it's an equal number. Whereas this equal number was not applied for Russian vaccine. I hope I get the time to talk about it, and Dr. Gonzalez, if you think my time is finished, let me know so I could go, address the Russian vaccine, okay? So you can see here, there are some limitations, okay? For example, we just started. Well, this was 2 weeks ago. I gave, same presentation in Italy and in Turkey. And you can see that we just started. So we cannot really assess, the safety and efficacy in general population. They already assessed it in their selective population, pharmaceutical companies. So we just started, and keep in mind, in children, we don't have any data, although Pfizer and Moderna is looking into children clinical trial. That is not going to be easy. That's very challenging for numerous reasons, Well, they recommend the vaccination in pregnant individual, but my question is, where is the data? Okay. They say, well, it's safe, blah, blah, blah. Fine. That's fine. We always care about risk versus benefit in clinical practice, but where's the data? Okay. And also immunocompromised person in all these clinical trials, different pharmaceutical companies, European, American, Chinese, Russian, Australians, they never addressed the issue in immunocompromised persons. So we really don't know how these vaccines, they behave in immunocompromised, patients, let alone, when it comes to transmission, we cannot assess it. Perhaps it takes us another 6 months, possibly in the middle of summer, to begin to accumulate and gather data on transmission rate. So we still have a lot, to learn from vaccine platforms, With respect to allergic reactions, usually those who develop, it's 4 hours after post-vaccination, and these are anaphylactoid rash or anaphylactoid reaction, usually epinephrine you must have, it's by injection, or there were 2 from my perspective. One I was consulting in the Detroit area, one in Miami area, was a full-blown anaphylactic rash, which necessitates hospitalization, oxygenation, epinephrine, steroid, and airways should be open. That's a different topic all together. And there are some people, they may develop, allergic reaction to PEG. So what is remarkable about Pfizer vaccine is really they tried the whole spectrum of population, the white population, Hispanics, okay, Africans and Asians, although the percentage of white was significantly high. I wish they put more weight, a little bit more on Hispanics, Latino, African Americans, and Asians. But overall, it's not a bad design. I mean, you can have some questions over the design, but this probably represents, United States demographic, population. And then you can see in terms of male, female, there were almost similar numbers. In other words, statistically, it's not very significant. Okay, so this vaccine is mRNA vaccine, and the stability, room temperature is 2 hours, and one of the issues of Pfizer is it requires -70, degrees refrigeration. That may not be practical or feasible for third world countries where resources are quite scarce. Let us say this probably can't be applied, let's say in Chiapas or in Amazon where I used to go, So again, these are some factors, that Yucatan Minister of Health needs to weigh in if they're going to adopt, vaccine. The second one is Moderna. You can see in Moderna, there are similar results as Pfizer, okay? So I have some objections over this cartoon. This should be actually the first dose. This should be second dose. They should have eliminated 2x here, 2x. Instead, they could have said first dose, second dose, which is 28 days apart. Okay, you can see, you know, there are some injection site adverse reactions, and even some people develop systemic adverse reactions. Again, Moderna, you can see the number of placebo arm versus vaccination arm. They're pretty much the same, okay? And again, that makes the interpretation easy for scientists, you know. Similar adverse reactions as Pfizer, okay? Again, they did a little bit better with respect to Hispanic, African Americans, and Asian, and other ethnicities compared to Pfizer, okay? And this is, by the way, the most expensive one. And after this would be Pfizer, which is Pfizer is $34. This one is $37, okay? And you can see there's a little bit difference, male versus female. So I don't know whether this was statistically different or not. I have no idea. So one of the things the Moderna did, and I really appreciate, they divided their population, okay, between 1655 and 70, and then 71 above, okay? So they used 4 different antibody assays to assess the pharmacokinetics of the antibodies. So they used an titer assay. So you can see in some individuals, advanced age, there's a little bit of, you know, downward, but it's not significant. However, they used different pseudo-neutralization assay. You can see that in this assay, okay, I explained the principles using luciferase enzyme. You can see that, you know, there is a waning of the antibody, but it did not reach that 50%, okay? When I extrapolated the data, it was around 54%, which is acceptable. You know, that Cmax and IC50 that you and I will learn in our pharmacology, you know, so at least it has 50% protection. So same thing, they used more advanced assay. One is PRNT MGN assay. There's one PRNT is a serological assay, you know, but in PRNT assay, you can see there were some of their patient cohort, they received vaccine, they had a sharp decline. But again, this sharp decline coupled with other, you know, parameters they used can be interpreted that still there is some levels of antibody protection post, you know, vaccine, in this case, post Moderna vaccine. Now, Moderna is 28 days apart, okay? Advantage of Moderna over Pfizer is it doesn't require -70, -20 is available all over Mexico, even Brazil, that I have connections with them, you know. Another advantage is it can stay in room temperature for 12 hours, whereas the Pfizer is up to 5 hours. So this seems to be more advantageous, you know. Now, this was a very nice study. Although it's a single study, I wouldn't pay too much weight on that, but at least, you know, it's beginning of our understanding of the dose interval. This study looked into so many patients. They received, you know, the vaccine. They found that with Moderna, the first dose provides up to 80% efficacy, whereas the other one, Pfizer, it provided efficacy up to 52%. So there's a big difference here between Moderna and Pfizer, okay? Again, this is a preliminary study, but again, it's telling us something is going on, which needs to be further validated by different studies. So in their data, they calculated the efficacy and interval. Certainly the Moderna tops, the Pfizer one. But what is very interesting is they're finding that after week 7, okay, after week 7, there's only 5% decline in antibody titer. So what it means is we don't have to apply Moderna, the second dose, on day 28. You can delay the second dose even up to 12 weeks, because if there's a 5% reduction, okay, week 8 would be 10%, week 9 will be 15%, week 10 will be 20%. That 20% overall is not going to change the profile of the efficacy. 20% is not going to change much, okay? So therefore, it makes more sense that we choose the second dose, let's say at week 10 or even 12, let alone this strategy can provide more vaccination, first dose vaccination to general population. But I also think that delaying Moderna's second dose can be beneficial to patients, because some patients, they develop acute reactions. Actually, some of them are really bad, okay, which warrants clinical attention. And so that's basically the issue. And I wonder myself, how these pharmaceutical companies, they came up with 21 dose interval for Pfizer and 28 dose. I'm sure they have some reasons, but I would like those reasons be published in the peer-reviewed journal, so we will have a better understanding of how they scientifically chose those intervals. But also, delaying the dose interval, that means delaying the immunogenic reaction. Let us say first dose really prime the individual, okay? The first dose, whether Moderna, whether Sputnik, whether any vaccine, it primes. However, the second dose doesn't prime here, okay? The second dose mission is to mount an immune response, to provoke an immune response, to induce an immune response. As we are all different genes, have different genetic backup, different immunogenetic pattern, different ethnicity, some of us may develop a faster systemic immune response, which may not be ideal. Yes, immune response second dose is normal. When patient has a fever, okay, it's normal, okay? But you saw it from some of those earlier slides. If that inflammation kicks in and goes higher and higher, that means vaccination effect turns into cytopathic effect, okay? So I want you to be aware of that as well.
Doctor,
so
we have
here,
10 minutes left.
10 minutes left. Okay, so let me go to Sputnik vaccine. Okay, this is a J&J vaccine. Okay, it's a single dose, 44,000 volunteers. You can see here, J&J vaccine, just Johnson & Johnson, okay? It's not as effective as a wild type, okay? Because there's a South African strain. There is also, other strains. So in South Africa, didn't produce as much good efficacy as it produced in USA or even Latin America. So that's an issue that the pharmaceutical company needs to address. The next one is Novavax. Novavax is an adjuvant proteins, okay? Uses matrix. The advantages matrix is that, it attracts, dendritic cells. As you know, dendritic cells, they have to signal B cells, to produce, antibody, So that's another one, but it's 2 injections and 21 days apart. And next month, I think, this will be in market in the United States. Now, They did a very good job on Novavax. They tested on wild-type virus, UK mutant, that's B117, and then South African mutant. You can see its efficacy has diminished in South African mutant. UK mutant is acceptable still, but it has diminished. So that's another thing, whether these vaccines can work on mutant viruses, or not, okay? So I'm gonna bypass these. Maybe we can have next month, another webinar on strains, because these strains are very good to understand clinically how you can interpret them. So I'm going to bypass these, and I'm going to go strictly to Sputnik. So here's Sputnik. I understand that your health ministry is considering to purchase it. Well, this was published on Journal of Lancet. By the way, in October, same journal, they did confirm that this Sputnik vaccine may be controversial, but they did say that they do not entertain any political discussion, okay? Then, this month, okay, February 2nd, they accepted the paper. This is a preliminary paper by the Russians. The vaccine is heterogeneous prime boost. In other words, complementary. Why complementary? Because they use two platforms, one viral platform. One is adenovirus 26, one is adenovirus 5, okay? So they induced, these recombinant technology in two adenoviral vectors, okay? All right, so this was a phase 3 trial, although when you read the paper, sometimes they say clinical trial 1 or 2. It's very ambiguous paper, by the way. If you want to take a note, you can find it, in the internet. Okay, so they did in Russian population. So here, the population is not heterogeneous. Okay, it's homologous population. Whereas, let us say, Pfizer, Moderna, they did white folks, African-American folks, Latin Americans, Asians. So you can see their clinical trial was strictly Russians. Although one can argue Russians, they are Abkhazia region, they could be non-white folks, but they did not break down, the race and ethnicity. Be it as it is, okay? So the first thing you wonder about this Russian vaccine is, why is it their vaccine arm has more than 16,000, their placebo arm a little bit over 5,000? Why is it? So these are confounders that are not in line with each other. So that can complicate the interpretation of data, let alone, okay, they broke down the age, very good, I like that, and they did antibody response, okay, in 2 different assays, I like that, okay. But why is it from that 16,427 vaccine arm, they only chose 342 patients to analyze their neutralizing antibody data? Why is it? Okay, they did not explain. Further, we knew that when these vaccine trials were going on, they were using either Chinese reagents or kits or Indian reagents. They do not have as much accuracy as European or American-made test kits. And that's true. As a matter of fact, at the beginning of pandemic here in the United States, we were using Chinese test kits. We had tremendous false positive, false negative results on PCR and even antibody test. Finally, CDC got into it, and now we have better, accuracy in terms of specificity and sensitivity. So that's the first thing came to my mind. Then, well, the time since first dose, they mentioned 80, but when you go into text of the paper, the median follow-up really was 48 days. So there is a big difference actually between 80 and 48 days, okay? The next concern that I have, they chose interferon, okay? Well, interferon is very variable assay. I can tell, I have a lot of experience on interferon, okay? Very variable assay. Plus, what type of test kit they were using, because interferon is a very sensitive assay, extremely sensitive. So my concern is over accuracy of these interferon assays, and you can see the interferon response was pretty variable in vaccine arm, okay? From the lowest, you can separate them. It would be one population, two population, and third population. So it was pretty diverse, let alone interferon response is an indirect indication of cytotoxic T cells. Further, I can add, at the beginning of infection, interferon pathway is shut down. And that's a huge scientific debate, whether at the level of gene expression is shut down or whether it is produced, but it is not the right interferon, goes after virus. So because of these, controversies, I think they chose the wrong, assay. They could have simply chose interleukin-4 or interleukin-10. That could be more direct result of the T cell function rather than interferon, given the discussions that we had so far. Interferon so far is very controversial. Then they classified the type of infection, high infection, medium, and general, but they did not take into account pre-symptomatic, asymptomatic, or super spreaders, and also, these workers, the investigators in Moscow. I've been in Moscow many times and St. Petersburg. I had clinical collaboration on tuberculosis and hepatitis with them, not anymore. So they commute by metro. So when they go to metro, were there any super spreaders there? Were there pre-symptomatic, asymptomatic? So there are some concern. And then when you look at the Th1 and Th2 arm, okay, well, the Russian vaccine did not address this arm. That is fine. But when it comes to cytotoxic arm, which is Th1 cells, they partially addressed it, and that is they only measured interferon, which may not be directly relevant to cytotoxic T function. So I'm going to stop here because I would like to get more questions on that.
Okay, thank you for your presentation. I am a little bit sad we didn't get to see all of that. They asked me to do a short briefing of what was said, but it's quite impossible. I'll just explain something in Spanish and then move on to the first question, okay?
Yes.
Pues agradecí al doctor Nassiri. Sé que muchos de ustedes están entendiendo la plática, pero me han pedido que haga una breve reseña de lo que se presentó. Lo encuentro muy difícil porque francamente hay mucha información. Creo que los puntos más relevantes que tenemos que tomar en cuenta para hacer la evaluación de los artículos que hablan sobre vacunas o de los resultados que se presentan acerca de la eficacia de las vacunas tienen que ser tamizados por los siguientes puntos, ¿no? Hay tipos de vacunas diferentes que funcionan, las plataformas que estamos revisando actualmente, por un vector viral o por RNA, y estas lo que hacen es que le dan información al cuerpo para que el cuerpo fabrique la propia proteína spike que va a servir para generar los anticuerpos. Y están las vacunas a partir de virus inactivados o las que nos dan precisamente la subunidad proteica o spike ya lista para que el cuerpo identifique. A diferencia de la infección, en donde hay una replicación constante del virus y entonces tenemos una cantidad suficiente de proteína para generar la antigenicidad o los anticuerpos, en las vacunas tenemos un número limitado de proteínas, del cual no se va a producir posteriormente más y por lo tanto la cantidad de proteína disponible sobre la cual se monta la inmunidad es menor. Ahora, entendiendo cuál es el mecanismo de la generación de las vacunas, lo que es importante al leer es cómo sabemos si la vacuna es eficaz y cuando un artículo nos está hablando de eficacia, exactamente cómo está haciendo los números. Y entonces nos llevó el doctor Nassiri sobre un repaso de qué estamos haciendo, el porcentaje de aquellos vacunados que enferman versus aquellos no vacunados que enferman, y con ahí se saca la matemática para determinar la eficacia potencial de la vacuna. Ahora, para hablar de eficacia tenemos que entender cómo es que ellos hicieron la definición operacional, porque nos pueden estar hablando de cuántas infecciones se previnieron, sean sintomáticas o no, lo cual se tendrá que hacer con estudios serológicos. O bien puede ser qué tanto previnió las enfermedades clínicamente evidentes, o qué tanto previno los ingresos a hospital, o qué tanto previno los ingresos a terapia intensiva, o qué tanto redujo el contagio, o qué tanto redujo la muerte. Y entonces para cada una de estos puntos de medición, la determinación de eficacia puede ser diferente. Y hay que entender que la vacuna puede ser útil desde el punto de vista de que prevenga muerte o infección grave y no necesariamente infección subclínica. Así que esto tendrá que ser tomado en cuenta en el tamizaje de las vacunas. Por último, hay que tomar en cuenta también la forma en la que la vacuna tiene que ser manejada para que llegue a los centros donde se realizará su aplicación, y el costo de la vacuna para que nosotros entendamos qué es factible en cada una de las unidades en donde trabajamos, porque una vacuna que requiere ultracongelación pues tiene dificultades técnicas para ciertos lugares de difícil acceso en función de otras que no lo requieren. Básicamente no quiero hacer el resumen de lo que él presentó porque es información muy detallada, sino que lo que estoy resaltando son los puntos a tomar en cuenta para que cuando uno se acerque nuevamente a los artículos tenga esta visión que el doctor Nassiri nos ha dado en función de los puntos a tomar en cuenta para entender lo que está pasando, independientemente de que creo que ha sido muy explícito en función de todos los cuestionamientos que se pueden ejercer acerca de la vacuna Sputnik en comparación con las otras, en los términos de cómo es que nos presentaron la información, cuál fue el tiempo de seguimiento de los pacientes, si midieron o no midieron la inmunogenicidad, cómo la midieron, qué tan heterogénea es su población y cómo todo esto incide en la interpretación final que tendremos que hacer de los resultados. So I did give a brief explanation, just guiding them on what are the pointers to consider when you're reading clinical evidence on vaccines. And then I want to address first a question you posed on. Por favor, las preguntas las podrán poner en el chat y las seguiremos seleccionando de ahí para presentárselas al doctor. Así que desde este momento, por favor, les invito a participar en el chat. You wondered whether the federal government is planning to vaccine indigenous population or population that's far away from cities. Actually, the decision is not made in the government of the state, it's made on the federal government. And then they are deciding, I'm not really clear, it's not really clear how they're deciding, but if they are deciding to move vaccination to smaller populations or in several states of the country, and then to those parts of Mexico City, which encompasses mainly 20 or 25% of the population of the country, to those areas who have more cases and just to manage the epidemic. But then they have also mentioned in media that the decision to vaccinate either in the city or on small villages depends on which is the vaccine available, which makes sense with what you said. If you had a vaccine that's not stable on the cold chain for a long time, then you would have to use it in cities or in centers in which you have access to ultra freezing. And if you have a vaccine that's more stable, then you can move that to population that's more distant to the center for vaccine application. So that's the answer to your question. And then I do have one.
Yes.
Do you expect variation in efficacy of the vaccines due to management of the product, mainly cold chain issues, especially those who need ultra freezing? I mean, I feel that you should be measuring as well the quality of the cold chain, and that has not been addressed in the papers I've seen, whether they are considering that even though in phase 3 trials the vaccine behaves in a certain way, once you account for the way the vaccine is applied or how it's transported, how well you kept the cold chain, this may also reduce efficacy of the vaccine.
Excellent question. Do you see this slide I moved? Do you see it?
I can see you.
You don't see the slide. Okay.
No.
Well, all right, that's fine.
Yeah,
I'm seeing
it's coming now. It's coming, I guess.
Yeah. Okay. This is what I was going to address, which relates to your question. Okay. This is a very important slide in the context of vaccine development. One is the raw material. Okay. The other one is some of these vaccines, non-mRNA vaccines, may require cell culture. Okay. And some of them is due to purification process. Okay. And then some could be environmental surfaces. Okay. In other words, when we make vaccine, there are so many variables, okay, not only in the context of technology, how technology is preparing the vaccine, but also variability from one batch to another batch. The only thing I don't see variable with mRNA vaccine, because they're very straightforward, okay, and you just put the code and deliver it into nanoparticle and produces the right protein. As a matter of fact, when there is all these variant that we are experiencing, UK variant, Brazilian variant, and South African variant, it is much easier for mRNA vaccines to go back in the lab in the matter of just 2 hours, not more than that, to change the genetic code and put it in the new nanoparticle and get vaccinated. Whereas other vaccines, they don't have that kind of luxury, you know, and simply. So they have to go back to lab, and it takes them, we calculate it takes them at least 6 months to develop new vaccine if there is a new, you know, strain of the virus. So far, we don't have a strain. We have only variants or mutants. But these are all possibilities. So your question is the right question, because in non-mRNA vaccine, you know, what are the contaminants? That's number one. And number two, what are the tools that you're using? Are those tools and technology contaminated, let's say with fungus? Because I had a case a few years ago, I was consulted, was a Brazilian in Sao Paulo area, you know, and he had fungal, you know, encephalitis. But then when we narrowed down on his clinical file, patient file, we noticed, again, this is no offense to any country, okay? I'm not here for politics. I'm here strictly to talk about science. Then the vaccine was made in Cuba. I've been in Cuba twice, excellent country. They're very good scientists, brilliant minds, you know. But when it comes to technology of making things, their technology level cannot compete with our technology, okay? So that can count for vaccine, you know, variability, because your question primarily was these variability that exist, you know. So, and even if you have the best, you know, recombinant technology, you need to prepare batch. You know, there are some variability from batch to batch, okay? So the other factor is, you know, the vaccination group, you know, whether they are trained to vaccine efficiently, which I addressed at the beginning of my presentation. And then we come to a point where everything is fine, but inadvertently, you know, the vaccine is thawed, you know, and may stay more than 5 hours, you know, and that can diminish the efficacy of the vaccine, okay? So there are so many variables, which goes back to your excellent question. And I cannot give you right answer right now, honestly. It takes time to come up with the right answer for you.
No, I know you cannot predict that, but I was wondering whether some essays were looking into that. But still, we have to wait and see.
No, we don't have essays addressing this issue, because, see, there are a lot of confounders in the questions you asked. There's so many variables. We don't have a standardized assay to address this issue. However, here is my point. If we develop a rapid neutralizing assay to assess, you know, what percent, I repeat, what percent of our serum after second dose has neutralizing antibodies. Again, I'm emphasizing on neutralizing antibodies, you know, and then couple that data with cytotoxic T cell function, which is not easy assay, which is not easy. Then that can give you a sort of direction, the future direction.
Okay. There's a question regarding whether, if you are aware or not of any candidate to vaccine or any vaccine in development that has the plans to be administered via aerosol, via...
Yes, yes, I'm aware of at least 4 of them. I'm aware of 4 of them, and actually 2 of them are being developed here in the United States and 2 of them in India.
Okay, so there are some
spray vaccines, yeah.
Okay. Is there any, you mentioned about the correct application of the vaccine in the deltoid muscle. Is there any report stating that maybe chronic consequences of wrongful application of the vaccine, such as chronic mild pain or something like that?
We don't have any report yet.
Okay.
Yeah, we don't have any report yet, but that's a very valid question.
Okay.
We don't even have report on immunocompromised patients. Okay. However, if I may expand on this, because I had one case of veganers, and I was consultant. You know, the patient was in Spain and had veganers and received the vaccine and ended up in hospital and developed anaphylactoid reaction.
Okay.
Vaganus granulomatosis.
I think you did address this before, but someone's asking whether we should be concerned about the vaccine being less effective against mutant strains. And could you just reaffirm this data?
Yes. Let me correct this person, this colleague. Let's not call it strain. Let's call it mutant or variant. Okay, because strain means biologically you have a totally different virus with devastating consequence. Okay, like influenza strain. So as far as mutants or variants are concerned, I wish I had more time because I had the slides to cover them. Maybe we can do it next time. We do not have any direct evidence that diminishing, let's put this way. I showed you a couple of slides on South African strain, you know, I mean variant. It's diminished, but still has some kind of protection. Okay, so at this point, no worries, clinically worries, because it gives you at least 50% protection. Now, you need to translate that 50% protection, okay, into clinical practice, hospitalization. Well, guess what? In case, let us say, severe form, that 50% protection brings you back to moderate stage, okay? Or if you were initially in moderate stage, that 50% protection brings you back to mild stage. Or if you are we're supposed to go to ICU, but you have 50% protection, it gives you to a non-ICU status. So still they're good.
So yeah, so it's a matter of how you define effectiveness, if you're defining it by infection or by severe infection or by ICU admission, right? So yeah. I wish that papers would present all these numbers on effectiveness on different definitions.
There is no paper
but
right
no
now.
one defines that way.
No.
And there's also a question that you did address before regarding vaccines, one-dose vaccines.
Yes.
You did present one, but my question to rephrase this one would be whether we have other one-dose vaccines being developed and soon to come out.
That's a very good question. The Russians are looking into that. I have some connections with them, and also Indians are looking into one dose. And you saw me, J&J, Johnson & Johnson is one dose, okay?
Yes.
And it is my understanding, indirectly I should say, that Moderna is looking into one dose, maybe by next year.
Okay.
Yes.
I think we have finished with pending questions. So I do thank you for your excellent presentation.
It's always a pleasure to talk to UADY. I consider Merida as my home.
Thank you. Agradecemos mucho a los asistentes que han estado hasta el momento con nosotros. Si hay alguna otra pregunta que quieran hacer llegar, nosotros estamos abiertos a que nos hagan llegar las preguntas por esta vía, porque el chat sigue activo, o de algún otro medio para que se las hagamos llegar al doctor Reza y podamos abordar algo más de la información que presentó. Esta presentación será grabada para su revisión posterior y sin más, pues agradecemos la presencia del auditorio y del doctor y les deseamos buenas tardes. Thank you, doctor. It was an honor to be with you again.
Likewise. Thank you so much. It's an honor for me. Thank you.